Hello to all of you. Let's discuss chapter number 12. And chapter number 12 and 13, they are connected chapters because these chapters are on cell division. So basically, I'm going to divide chapter number 12 and 13 into three different parts. And in the first chapter, first part, I'm going to talk about cell division called mitosis. I'm going to go over the steps of mitosis, and then I'm going to talk about the regulation part. And then in the third part, I'm going to discuss chapter number 13, which is all about the meiosis. So first of all, before we start talking about the cell division, I would like to show some pictures. And so you can see here, so basically a small elephant grows up and becomes a big elephant, right? And you must have seen sometimes some of the animals like especially like snakes and frogs, they shed their skin and then they get the new skin. And third is our life starts at the conception point when the sperm goes and fertilizes the egg. So when the sperm goes and fertilizes the egg, it becomes the fertilized egg, and the fertilized egg, it actually divides and that turns into an embryo, and then after a few months a baby is born, and then the baby goes through different stages, and then our life's journey starts and it goes through all these stages, and we become adults, right? So we become big. So our life basically starts from this fertilized ovum. So our life basically starts from this fertilized ovum, and then we go through different stages and we become big. So the question is how all these things. So I've shown you three pictures: a small elephant becoming big elephant, animals shedding the skin, and then they get the new skin. And our life starts as a fertilized egg from basically one cell, which is a fertilized egg, which is because of the fertilization of the sperm with the ovum. So those are two gametes. So we go through all these stages. We go through infant stage, toddler stage, and then we become big. So how does this all goes? So how does it go? All goes? The answer is cell division called mitosis. So cells constantly reproduce exact duplicates of themselves. Cells constantly reproduce exact copies of themselves, and they reproduce exact copies of themselves for growth, for repair, and for replacement. And the cell growth and the division happens in a series of recognizable steps called cell cycle. So basically our life is, so in these two chapters we are going to talk about cell division, and that, as I just said, there are two kinds of cell divisions. I would like to explain here that our life is basically going through two different kinds of cell divisions: mitosis and meiosis. So as I said, our life starts from a fertilized egg, and then that cell keeps on dividing. It becomes an embryo, it goes through different stages, then a baby is born, then the baby becomes big, it becomes a toddler. So all these things, so we are growing and developing, it all happens because of cell division called mitosis. And then when we reach to adolescent age, puberty age, then we start making our own gametes, and gametes are formed by a cell division called meiosis. So these gametes are produced in us, and then two individuals of different sex who produce different gametes, like females produce ovum and the males produce sperms. Then sperm fertilizes the egg, and then the new life starts. And then that fertilized egg goes through a cell division called mitosis. So this is the way whole life's journey is going on. So the life is going on because of two cell divisions called mitosis and meiosis. So the cell division called mitosis, it's a very, very important feature of this cell division, that it results in two daughter cells which are genetically identical to each other. So in the cell division called mitosis, from one cell, from one cell, two cells are formed, two daughter cells are formed, and they're genetically identical to each other. So here is a very, very important, remarkable difference between mitosis and meiosis. So in mitosis, from one cell, two cells are formed and they're genetically identical to each other. In the cell division called meiosis, from one cell, four cells will be formed and they will be all unique. They will be all different. So this is a very, very important difference between mitosis and meiosis. So before we go and talk about cell division, I would like to explain some terms because they will be helpful to understand. So somatic cells, what are somatic cells? All the cells in our body, they're called somatic cells. And the sex cells, the gametes which are produced in the reproductive organs, they're called gametes. So all the somatic cells, all the cells in our body, they divide by a division called mitosis. And the sex cells or the gametes, they divide by a division called meiosis. And now I would like to talk about our genome and how it is present in our nucleus. So genome basically means all the DNA in a cell constitutes the cell's genome. Okay, and this DNA is wrapped over proteins. Okay, now here are two very, very important terms. One is chromatin and another is chromosomes. So I want all of you to understand what chromatin means and what chromosomes means. Now, as I said, the whole DNA in our cell is called the genome, and this DNA is wrapped over the proteins. So when the cell is not dividing, at that time, this DNA, which is wrapped very tightly over the proteins, forms a dense mass and it is called chromatin. So when the cell is not dividing, at that time, the DNA is wrapped over proteins, but it is in the form of a dense mass and it's called chromatin. When the cell starts dividing, then this chromatin gets condensed and it takes the shape of structures called chromosomes. So this is something very, very important to know, the difference between chromatin and chromosomes. So you can see here, as I said, DNA is wrapped over histone proteins. So the DNA is wrapped over histone proteins and it forms a dense mass called chromatin, and then this chromatin gets condensed and it takes the shape of chromosomes. So this is all what I just explained. So DNA is located in the nucleus and controls all cell activities, including cell division. So in the non-dividing cells, this long DNA wrapped over proteins is called chromatin, and when the cell starts dividing, it takes the shape of chromosomes. And the structure of chromosomes is like this. It has, now this is a duplicated chromosome. Okay, so it's important for you to understand what a duplicated chromosome is. We are going to talk about cell cycle very shortly, and in that cell cycle I'm going to explain you that how DNA duplicates itself. And when the DNA duplicates itself, and when that DNA condenses, it takes the shape of a chromosome like this. So if the DNA is not duplicated, then the chromosome will have only one arm. But when the DNA gets duplicated, the chromosomes get this kind of a structure, and this is a duplicated chromosome. So which has two chromatids, and so this part where these two chromatids, so this part where the two chromatids are in very close contact with each other, this region is called centromere. So these arms are called chromatids and this part is called centromere. So this is a region where the two chromatids, they are in very close contact to each other. That's called centromere. And as I said, this duplicated chromosome will be visible when the DNA has been duplicated. We're going to talk about the stages of interphase in which the duplication of DNA takes place. So basically, because the DNA duplicates, this chromatid is actually a copy of this chromatid. So these two chromatids, they are called sister chromatids. So this is just a cartoon. It's showing that the chromatin is like this and it condenses and takes the shape of chromosomes. So this is all what I explained. So this is, as I said, when the DNA is not duplicated, it's like this, and when the DNA gets duplicated, it takes the shape like this. So this is centromere and these are the sister chromatids. So this is the centromere and these are the sister chromatids. So basically, this is overall showing what happens in mitosis. There are many other things which takes place in mitosis, but this is overall. So you can see this is the DNA. It gets duplicated and it takes a shape like this. And then, so these are two sister chromatids. They're copy of each other. They're clones of each other. And during the cell division, these two sister chromatids, they separate. Okay, so basically, during the cell division, in the cell division called mitosis, basically, first of all, there is a division of the, basically the name mitosis is given for the cell division called mitosis. So, but mitosis actually word means division of the nucleus. Okay. So we will be talking about cell cycle. In that cell cycle, you will see that the cell goes, what is the cell cycle? Cell cycle is, actually let's talk about this first. Okay, this is cell cycle. Now what is cell cycle? Cell cycle is the sequence of events through which the life of the cell passes. Okay, cell cycle is the sequence of events. So cell cycle is the sequence of events through which the life of the cell passes. So first and the longest phase is interphase. Okay, interphase. Okay, this is the longest phase. And second phase is mitotic phase. Now interphase is a phase when the cell is actually not dividing, but in the interphase, what the cell is doing, it's actually preparing for the upcoming division. Okay, it is duplicating everything, it's preparing. And mitotic phase is a phase in which the cell is actually dividing, and mitotic phase has two subphases. First is mitosis and second is cytokinesis. Mitosis is the division of the nucleus and cytokinesis is the division of cytoplasm. And you can see here, this phase, this all phase is interphase. Okay, going from here to here, this is all interphase, and this phase is mitotic phase, and this phase is mitotic phase. Mitotic phase has mitosis and cytokinesis. So this is the time period of the cell. So this is the time period in the life of the cell when the cell is actually doing preparation. Okay, so cell spends a lot of time in preparation, and once the preparation is done, then the cell divides. So first of all, the nucleus will divide, and that is called mitosis, and then cytoplasm divides, that's called cytokinesis. The interphase has three subphases, G1, S and G2. Now what is G1? G1 is a phase in which actually the cell is duplicating all its organelles. In G1, the cell, or it's called gap 1, in this cell is duplicating all its organelles. And what is S phase? S phase is that phase in which the cell is duplicating its DNA. And G2 is that phase in which the cell is making some proteins which will be required for the upcoming division. So you can see how much time the cell passes in the interphase or in the preparatory phase. So here it is in the G1 gap 1, it is actually duplicating all its organelles. Here it is duplicating its DNA, and here it is preparing some proteins which are required for the upcoming division. And once all the preparation is done, the cell enters into mitosis, where the cell actually divides its nucleus, and then cytokinesis. In this phase, cell actually divides its cytoplasm, and this is called mitotic phase. Now the mitosis, which is the division of the nucleus, has 5 stages: prophase, prometaphase, metaphase, anaphase, and telophase. Now you can see here, this is the G2 of interphase. So in the G2 of the interphase, which is the last phase of the interphase, you can see here the cell has duplicated everything. Okay, so in the S phase, the DNA is duplicated. So this is the cell is in the interphase. It is in the G2 phase of the interphase. Here, because the cell has passed through S phase, the DNA has been duplicated. So this is a chromatin. So this is a chromatin. Okay, so this is a chromatin, and this chromatin has duplicated DNA. And you can see here these are centrosomes. These are centrosomes, and they have also been duplicated. So this is G2 of the interphase, means this is the last phase of interphase. The cell is all prepared. The cell is all prepared for the upcoming division. Then the cell enters into prophase. So so there are 3 most important things which takes place in prophase of mitosis is, first of all, this chromatin is going to get condensed, and it is going to take the shape of these chromosomes. So chromatin gets condensed, and it takes the shape of chromosomes. The nuclear membrane, it gets fragmented, it gets broken, and then these centrosomes, they start moving apart, and they start forming the spindle. I'm going to discuss what spindle is, what it is made of, and what is how the spindle is formed, and what is its function. So this is what is happening in prophase. 3 important things happening in prophase. Number 1, chromatin gets condensed. It takes the shape of chromosomes. And the nuclear membrane, it gets fragmented. And these centrosomes, they start moving apart. The centrosomes, they start moving apart, and they start forming the mitotic spindle. So in the prometaphase, in the prometaphase, basically what happens is this spindle apparatus is forming, and these mitotic spindles, they actually start attaching to the chromosomes. Okay, so in the prometaphase, these centrosomes, they have reached to the 2 different poles. The centrosomes, they have reached to the 2 different poles, and from these centrosomes, I'm going to explain you. These centrosomes are also called microtubule organizing center, and the mitotic spindle is made of microtubules. We have already talked about what microtubules are when we talked about the cytoskeleton, and at that point we also discussed that these microtubules, they form the mitotic spindle. They form the mitotic spindle in the mitosis, and they help in the separation of the chromosomes. So in the prometaphase, these centrosomes, you can see here at the end of the interphase, there are duplicated centrosomes, and in the prophase they start moving apart, and they start forming the mitotic spindle, because these centrosomes are microtubule organizing centers, and from these centrosomes, the microtubules, they arise and they form the mitotic spindle. And when the cell reaches to prometaphase, you can see here how the mitotic spindle is forming, and these microtubules, which are originating from these centrosomes, they are attaching to protein complexes called kinetochores, which are there which are associated with the centromeres of the chromosomes. So this is what is happening in prometaphase, and in the metaphase you can see all the duplicated, and important thing to understand here is the chromosomes are all duplicated, because I told you here the chromatin is all duplicated. The chromatin is all duplicated, and when these chromatin gets condensed, it takes the shape of chromosomes. These chromosomes, they are all duplicated. So if you see a chromosome with 2 arms, it means it's a duplicated chromosome. So this is important, that in the metaphase all the duplicated chromosomes, they have come in the center. And in the anaphase, the sister chromatids, they are getting separated. So in human beings we have how many chromosomes? 23 pairs or 46 chromosomes. So in the humans, in the mitosis, all the duplicated 46 chromosomes, they come in the center. And in the anaphase, it is the separation of the sister chromatids. So which means that all the 46 chromosomes, all the 46 duplicated chromosomes will be having sister chromatids. Every duplicated chromosome will have 2 sister chromatids, and every duplicated chromosome has 2 sister chromatids, and in the anaphase it is the separation of the sister chromatids. So which means that 46 sister chromatids go this side and 46 chromatids go this side. 46 chromatids go this side and 46 chromatids go this side. And next phase is telophase. In the telophase, it's basically in the anaphase, actually the cytokinesis, which is the division of the cytoplasm, starts. So in the anaphase, the division of the cytoplasm starts. It initiates here. So in the next phase is telophase. You can see how the cytoplasm is getting separated, because cytokinesis starts here. And basically around the so on this side 46 chromatids reach, and on this side 46 chromatids reach. So around these 46 chromatids, there is a development of the nuclear membrane. So there's a development of the nuclear membrane on this side, there's a development of the nuclear membrane on this side. And basically the mitotic spindle, it all disappears. So this is what the telophase is. So telophase gives the appearance of one big cell with 2 nuclei. So in the telophase, the cell looks like one big cell with 2 nuclei, because here the cytokinesis is not completely finished. And the nuclear membrane is appearing, and all these things are happening. So it gives the appearance of one big cell with 2 nuclei. That's what is telophase. Okay, now I'm going to talk a little bit more in detail about the mitotic spindle. So as I said, from the centrosomes, I'm going to explain from here. These are the centrosomes. So in the interphase, there's a duplication of centrosomes also, because in the G1, which is the gap 1 phase, the all the organelles, they get duplicated. And centrosomes also get duplicated, and they move to the 2 different poles. And from the centrosomes arise the microtubules, because centrosome is called the microtubule organizing center. So you can see all the microtubules are originating from the centrosomes. Okay, and these are the duplicated chromosomes. At the centromere, there are some protein complexes associated. With the centromeres, and they are called the kinetochores. There are some of the microtubules which attach to the kinetochores, and those microtubules are called kinetochore microtubules. And there are other microtubules which do not attach to the kinetochores. They are called the non-kinetochore microtubules. So they basically help in the stretching of the cell. So this is whole mitotic spindle, which includes kinetochore microtubules, non-kinetochore microtubules, and also there are small microtubules which originate from centrosome, and they're called esters. So the spindle includes centrosomes, spindle microtubules, and the esters. An important thing to understand here is basically, so as we just talked about, the kinetochore microtubules, they attach to the kinetochores. Kinetochores are the protein complexes associated with the centromeres, and non-kinetochore microtubules from opposite poles overlap and push against each other, elongating the cell. So these chromatids, these sister chromatids, they are attached to each other by a protein called cohesin, and in the anaphase, this cohesin is degraded by another enzyme called separase, and which helps in the separation of these chromatids. So in anaphase, cohesin is cleaved by an enzyme called separase. So cohesin is a enzyme, it's basically a protein which helps in the binding of the sister chromatids together. And cytokinesis, so we have already discussed the stages of the mitosis, followed by this telophase, or actually important thing to know is cytokinesis actually initiates at anaphase, then it continues in telophase, and then it continues after telophase also, and then eventually the cells, they divide and form two daughter cells. From one parent cell, two daughter cells are formed. Cytokinesis occurs by basically cytokinesis, which is the division of the cytoplasm. It takes place by two different processes in animal cells and the plant cells. It's different in animal and plant cells. In the animal cells, it is cytokinesis occurs by the formation of a cleavage furrow, and in plant cells, this is the formation of the cell plate. So in the animal cell, this is the way the cleavage furrow is formed, and it is formed by the some proteins. It basically there's a formation of a contractile ring of microfilaments, and microfilaments, they're formed of some proteins, and then the daughter cells are formed. In the plant cells, there's a formation of a cell plate between the two cells. The two cells are formed by the formation of the cell plate, and this cell plate is formed by the fusion of the vesicles, which are formed from these vesicles are secreted from the Golgi apparatus, and then they come here and they basically fuse and they form the cell plate, and that separates the two plant cells, because in the plant, from the cell plate is formed the new cell wall, because remember that plant cells have a cell wall, animal cells do not have the cell wall. Okay, after this part, we are going to discuss about the regulation of the cell division, of the cell cycle. I'm going to make another video for that.